Good morning, everyone. All right, just waiting for a few more people to roll in. All right. So, aim today is to pick up on what we're talking about in terms of gene regulation and also to start looking at examples, right? So, the two examples we're going to use for prokaryotes, and prokaryotes have sorry, here on the mouth, pretty cool way of controlling their gene expression. Let me just get to this is kind of where we were kind of ending up essentially last class. So one thing that I want you to do as best as possible is to try and keep in mind the context, right? Because otherwise this is just a bunch of words and, almost mumbo jumbo essentially, right? And really it's a very, very cool process by which organisms, control how they are. And so in prokaryotes, this is actually really cool because essentially, if you think about bacteria exist, when they're not in the lab, in, an environment with lots of other organisms around them, right? There are, even in your gut, there are potentially hundreds of different species of bacteria that colonize different parts of your intestine. And certainly in the soil and other environments and water and so on, there are lots and lots of different species. And so all these species are in this constant arms race and competition to succeed and grow and reproduce faster than anyone around them, right? And so it's just like a giant, cage fight essentially in terms of bacteria and other organisms too. And so bacteria have to be able to respond really effectively to changes in conditions and changes in nutrient availability so that they can capitalize on those changes or minimize their effects if they're unfavorable, but do no more than is absolutely necessary, right? Because if they overshoot the mark and produce more proteins than they need to, then essentially that has an energetic cost, which means they're going to grow more slowly than the people, well, not the people, bacteria around them in their environment, and they're essentially going to lose that arms race, right? So a lot of bacterial control of gene regulation, which in bacteria is largely control of transcription, is all about kind of maximizing the return, minimizing the cost, right? So they are very, what I would call cool word, parsimonious, right? And that means like they're very careful with their money, essentially. And in bacteria, money is basically ATP and GTP, right? It's energy. And so what I want to do first is give you some kind of terminology and ways of thinking about this. And then we're going to go into the first of two examples, which is going to be the lac operon, right? Which is a relatively straightforward, but a really neat example that's kind of going to get you into that kind of thinking frame, right, of what we're talking about. Just going to make sure I can bring up the chat in case anyone asks any questions. All right, so essentially when we talk about control of transcriptional regulation, which isn't, again, a large part of what goes on in bacteria, we're talking about the outcome, right? We're not talking about the action of the thing that's going on, but the effect that it has on transcription. So negative regulation, really you're saying no to transcription, right? And so you're turning off transcription by using a protein that inhibits transcription, represses it. That's why it's called a repressor. So if you're turning it off, then essentially the default state is always on. And that's very typical of bacteria. Eukaryotes, typically it's more positive regulation, which is the opposite. So the transcription is typically off and then you have to turn it on, right? So it's a positive thing. You're getting transcription out of that gene, right? And that's done with a variety of different proteins, transcription factors and transcriptional activated proteins. Okay, so we have turning things off is negative regulation. Turning things on is positive regulation. And so all of this is done, and I want you to kind of really keep this in mind. These actions, pretty much all of them really, are carried out by proteins that bind to DNA, right? They have a particular shape, and that shape can change, but a particular shape which recognizes a short sequence, 10, 20, 30 base pairs maybe, of DNA, right? And that sequence is called the activated binding site, or just the DNA binding site for that particular protein. So it won't bind anywhere else. It's not going to bind over here. It's not going to bind up here. It's going to bind where that DNA sequence is found. And that DNA sequence isn't necessarily present in front of every protein. In fact, it's typically not. It's typically in front of particular proteins. Okay, so remember, these DNA binding proteins, they bind a specific sequence in DNA that they recognize, right? That's where a lot of the specificity of the control of gene expression comes in. So moving on, we have a couple of extra wrinkles, right? So we have inducible transcription, where something can induce that repressor to leave. So that repressor will bind in the absence of that inducer, and it will turn off gene expression. Right, so when something isn't around, whatever that might be, gene expression is on. You need the presence of that something, again, whatever it might be, to turn it on. And that turning on is done by removing something. That's different from positive regulation in a sense, right? If you think about it, because positive is all about adding something to turn on gene expression. Inducible transcription is about removing it. Right, so essentially something's blocking, right, the path of the transcription machinery, and you have to have that inducer present for it to leave and clear the way. And so quite commonly these are metabolites, not always, but quite commonly they're metabolites, right? So they're things, lactose, for example, is a great example, which when present will result in the transcription of that particular gene or operon. The opposite is repressible, right? So in a sense, this is like negative regulation, right? So the default here is on, but here you're trying to turn it off, but you turn it off in the presence of a co-repressor, right? So this is a little bit different. I mean, it's kind of same outcome, but a slightly different wrinkle to it, right? To negative regulation. Because now it's not just any old protein, it's a protein and something. Again, typically a metabolite, right? So here we have the repressor, and that repressor is called an aporepressor. So let me bring up the chat. So an aporepressor is a repressor. Oh, it looks weird using an O there. Without its co-repressor. And so that results in no binding to its DNA target sequence. Only when that co-repressor is present does the aporepressor adopt the right shape and bind to that DNA sequence. Anybody got any questions so far? Things are going to get wild. And so that's kind of quite neatly shown here. In inducible transcription, both of these have repressors. The difference is that the inducer is kind of the similar in a way to a co-repressor. It's something that has to bind to that protein. But in this case, the inducer induces it to leave, right? So it changes its shape so it doesn't bind. Whereas in repressible transcription, it does change its shape, but then it will bind, right? So the same idea, we've got a two-part system, protein and something else, protein, something else. In inducible transcription, when those two things are together, you have transcription because it is induced to leave. In repressible transcription, when you have those two things together, you don't get transcription because now it's in the correct shape to bind and block RNA polymerase from moving down the way. So another little wrinkle is that you can have something called autoregulation. And so essentially, if this gene or operon encodes something which produces the metabolite that binds to the repressor, either this one or this one, the product of this gene in terms of the metabolite can regulate the expression of the gene. So if you want a gonzo amount of stuff, you might have a case where you have a positive autoregulation, right? Whenever more of a particular metabolite for whatever reason. Maybe you only want to maintain a certain level of metabolite, in which case you may have negative autoregulation. So the product down here, kind of that away somewhere, once it reaches sufficient levels, turns off transcription of the things that make it. Right? Also, negative regulation is typically more common in prokaryotes. In prokaryotes, most genes are normally on unless they're actively turned off. Right? Whereas positive regulation is more common in eukaryotes. So in eukaryotes, like you and me, most genes are normally off, and then we turn them on when we need them to, right? So it's a very different approach, essentially, right? It's both have their pluses and minuses. Okay, a little bit of a quick reminder, right? Gene activity and transcription is regulated through control of transcription, as I said already. And we have a couple of funky things in bacteria, right? So in bacteria, all of the structural genes, which you can see here, that are involved in a particular process, production of product Y, let's say, are grouped together along with their regulatory sequences in something called an operon. So an operon contains structural genes and then a couple of regulatory regions, one, the promoter, which is where RNA polymerase binds, and two, the operator, which is where the regulator binds. These are just sequences of DNA, pieces of DNA that have particular sequences, right? Remember, we've got the -10 and -35 sequences in our promoter. And here, so the idea about this is really this, anything that binds here prevents RNA polymerase from transcribing these structural genes. It either does it by physically blocking RNA polymerase from kind of moving through, right? It's like a big concrete bollard in a road, you know, whether you like it or not, you're not getting through. Or it prevents RNA polymerase from binding. So it kind of occupies the space that RNA polymerase would normally occupy. And so you can't have two things there at the same time. So RNA polymerase does not bind. So that's one thing, operons, right? The second is those structural genes are transcribed in one polycistronic mRNA. Each of these can be translated separately into proteins, but the mRNA for all of these genes is in one large, long mRNA called a polycistronic mRNA. So the big plus side of all this is that, hey, you want to make product Y because precursor X is present. So you need all of these proteins to do so. So you group them all together genetically in the DNA, and you control all of them at one shot. So this is called coordinate regulation. And it's a very efficient way of making sure that, hey, if you want to use this particular metabolite, well, you produce all of these proteins that you need to do so, whatever they may be. And then the extra bit, right, and this doesn't necessarily have to be anywhere nearby. It can be, but it doesn't have to be, is another gene. This encodes a regulator. Which is a protein that can, under right conditions, bind to the operator. And those conditions are obviously going to be specific to, you know, whatever operon we're talking about, and they do vary. So these are the general components of a regulatory system in bacteria. Operon, which consists of regulatory regions and structural genes, and then regulator gene somewhere else, and the regulator protein. Okay, give you just a second to cogitate on that. Anybody have questions? Because we're going to get serious. Operons. Operon is a group of structural genes and their regulatory regions that control them, all grouped together. The operator, right, which is this bit in red here, is between those structural genes and the promoter. So anything bound in the middle between the two prevents the RNA polymerase moving through and, you know, making that polycistronic mRNA. So that operator is where it is for a very specific reason. Anything bound to it prevents RNA polymerase from doing its job, which is to transcribe the polycistronic mRNA for these structural genes. Cool. Good question. And please do ask questions all the way through, because I know that this particular topic is one which students tend to struggle with, not because it's beyond their abilities, but it's more that you're being asked to think in ways which you're not used to thinking, right? And there's a lot of stuff going on, which you would expect, right? No one expects gene expression to be simple. But please do ask questions, right? I don't expect you to get all of this immediately. It'll take time, and now's a good time to ask questions. Friday's a good time to ask questions too. Monday, Wednesday, Friday, you know what I mean. But anyway, do ask questions. So we're going to use two examples to look at this, and they do very different, they're controlled in different ways. And so one thing that I want you to get from these two examples is that bacteria have kind of two modes of controlling gene expression, right? They've got a yay, we're going to make something kind of mode, right? Gonzo mode is kind of what I call it, right? And so they can increase the transcription of a gene 100 or 1,000-fold. It's like what I think of, in what is it, Frankenstein's monster, when they have that giant switch, like that they go like that to connect the lightning and resurrect that kind of patchwork guy called Frankenstein's monster. Anyway, it's kind of like that, right? So it's this big change. Let's turn on gene expression. But also, bacteria have to be careful because you don't want to make too much of something that you don't need. And so they've also got, like small guy in a baseball cap in a corner with a dial, like kind of tweaking the level of gene expression, right? And that's a much finer control, right? It's usually about 10-fold difference between the dial all the way to the right and all the way to the left, right? No spinal tap here. There's no 11, right? But actually, I have to watch Spinal Tap. I keep meaning to. It's like part of my cultural education. But there's those two different aspects, right? We're going to see examples of different ways of doing that. Remember the big on, 100 to 1,000-fold change in transcription, and the little on off, which is more, 5 to 10-fold. So they can tweak it essentially. Okay, so we're going to talk about the lac operon, and basically what we care about is this, right? So lactose is a disaccharide, and it consists of a glucose and a galactose. I'm just thinking of Battlestar Galactica right now for some reason. Anyway, so essentially, bacteria swimming along quite happily in its environment. It needs to turn glucose into ATP to be able to do all the other stuff it needs to do. Even just swimming with its flagella takes an astonishing amount of ATP. How much was it? Like 30 to 40 ATP for one single rotation of its flagellum, which is nuts. Anyway, so basically bacteria have to be kind of pretty careful, right? They have to get enough energy, but they also want to get the energy in the easiest way possible. So if glucose is present, why would you go through all of this to generate more glucose? You just use glucose. But if glucose is not present, then even though this is a bit of a fiddle fart to get, you still get glucose out of it at the other end, right? And so the lac operon is all about controlling the production of stuff, this lactose permease, which gets it into the cell, and this beta-galactosidase, which turns it from lactose into galactose and glucose. You want to control that so that you don't produce it when glucose is present, but you produce it when lactose is present and glucose is not. Obviously, if glucose and lactose are not present, there's no point producing it, right? Because there's nothing for you to turn into glucose. So this kind of like balancing act, right? What do I have? What do I need? What's the easiest way to get from what do I have to what do I need, right? This balancing act is part of how the lac operon is controlled, right? So the lac operon consists of 3 structural genes, Z or Z if you're American, Y and A. Z is what is it? Oh yeah, that is I should know this. Beta galactosidase, right? So that's this enzyme here. Lac Y is the permease, right? So this is a transmembrane transport protein, which gets lactose into the cell, but nothing else. And there's another one as well, which isn't very clear exactly what its purpose is, but it's a transacetylase. Don't have to worry about that. That's lac A. So those are the 3 structural genes, Z, Y, and A. And we also have our promoter, our operator, so that's lac P, lac O, and a repressor gene called lac I. And this is all kind of put in here. And so in the absence of lactose, allolactose, that lac I repressor binds to the operator. So if you don't have any lactose around, there's no point producing the stuff which lets you turn lactose into something useful, right? It's be a complete waste of energy. So in the absence of lactose, this whole operon is off, right? So this is negative regulation. And this is turned off by this repressor protein here called lac I. Right? Any questions? And you can also see what I was talking about earlier about why lac O is important, like where it is. This lac I repressor covers the promoter. So when it's bound, RNA polymerase is not getting in here. Another thing worth pointing out too is that this repressor protein is always produced. So that's something that's called constitutive. So the production of lac I doesn't rely on anything else, right? It is just always on, never gets turned on, off. Oh, I'm sorry. I'm not getting a good night's sleep right now for some reason. Okay, so our inducer is lactose. So when lactose is present, now we have lactose binding to the repressor protein and either preventing it from binding to the operator, or if it is bound to the operator, causing it to disassociate, right? So it's no longer physically able to bind to that bit of DNA. And so now that this is removed, right, it's induced to leave, RNA polymerase can access the promoter. It will transcribe that polycistronic mRNA, and it will generate these 3 different proteins. Right, so so far, it's relatively simple. So this is an inducible expression system. Okay, now we have an extra wrinkle. So this is the large-scale change of expression of the lac operon. So removal of the LAC-I repressor protein in the presence of lactose, changes the transcription of this operon by a very large amount. But if you remember, I was saying earlier, what if glucose is present? So if you're in the presence of both lactose and glucose, it would be energetically much, much more favorable to use glucose, right? Because even once this lac operon stuff has done its job, you still have to break glucose down and generate ATP from it. Right, so essentially you're paying for an extra couple of steps, import of lactose, generation of galactose and glucose, that you don't have to pay for if you have glucose. So really what you want to do is kind of control how much of this lac operon works if glucose is present. And again, don't forget, the whole point of this is to generate glucose. So once glucose levels start rising sufficiently, either because glucose is present or lactose is being turned into glucose, you can dial things back a bit, right? Because, I mean, it's not like bacteria can get diabetic, but they can only process so much glucose at any particular time. So the more they have, it's just going to end up being hanging around surplus to needs. And so this is where the fine control of the lac operon comes in. And essentially, we're going to figure out how much, or the bacteria does at least, how much glucose is present, right? And we don't do that directly, right? So actually, we don't either, right? Organisms detect the level of glucose by another compound. That compound is called cyclic adenosine monophosphate, or CAMP, or CAMP, right? So this thing is essentially our metabolic sensor, and humans use it too, not just bacteria. And so and essentially cyclic AMP levels are inversely proportional to glucose. Let's write that down. So basically when glucose levels concentration are high, CAMP levels are low. When glucose levels are low, CAMP levels, oops, if I spell it right, are high. And it's a teeter-totter, basically, right? It's a, oh shit, I can't remember the other word for it. Mine is blank, right? So essentially, glucose, cyclic AMP kind of does this, right? And so really you're not detecting glucose levels, you're detecting their effect on cyclic AMP levels, right? And that's what bacteria do to control the lac operon. And so they do it via something called the catabolite activator protein, CAP. And so when you have lots of cyclic AMP, you will have it bound to lots of catabolite activator protein. And this basically promotes the transcription of the lac operon, right? So this is positive regulation. And again, stop me at any point, right? So essentially, and this is CAP is produced by, you know, um, another protein, another gene, sorry. And when lactose is present, right, because otherwise RNA polymerase isn't going to bind, the presence of cyclic AMP allows CAP to promote the binding of RNA polymerase. I don't know if that means it stabilizes it more or better on the promoter or recruits it to the promoter. I'm not entirely sure. But whichever it is, it increases the transcription of the lac operon by about tenfold. So again, when cyclic AMP levels are high, it binds to the CAP protein, and that complex, the CAP-CAMP complex, promotes transcription through increasing the efficiency of polymerase binding. And so you get more lactose turned into glucose. But you're like, we have more glucose present, right? Even if we don't have glucose present externally, we now have glucose present in the cell. And so now glucose is going to start going up, right? Obviously, if it's needed, it's going to get turned into something else very quickly. And so it's not going to go up quite as quickly. But as soon as glucose levels start going up, CMP levels start going down. So when CMP levels decrease, there's less of this CAP-CAMP complex. And when there's less of that, RNA polymerase doesn't bind as efficiently. It still does, just doesn't do it as well. And so transcription occurs, but it occurs at a lower rate. So this is that dial, right? It's about, from what I've read, about tenfold difference, right? So if presence of lactose increases the transcription of the lac operon by say 100-fold, but when glucose is present, that drops by tenfold. Now you only have a tenfold increase in expression, right? In the presence of lactose and glucose. So you can see that this is a very, very neat way of kind of basically kind of juggling that balance, right? You know, we've got something that we can use to generate energy, but it takes energy to get it, right? To get it in a form we can use. So as soon as we start producing something that we can use for energy, we want to just kind of like dial it back, right? Because all of this whole process takes energy, right? And that energy has to come from somewhere. It's coming from glucose, essentially, right? So you don't want to do any more of this than absolutely necessary to get as much of that glucose as you need for whatever metabolic processes you have going on. So as soon as glucose levels start increasing, you know, either because there's now glucose present in the environment, or more likely because glucose is being produced from breaking down lactose. As soon as those levels start going up, that dial starts getting turned down, right? And so basically you're trying to match supply and demand, basically. You know, the supply of glucose versus the demand for glucose. And the better you can kind of match that, you know, closely, the more successful you'll be as an organism, and so the more likely it is you'll pass on your genes to your offspring. Okay, questions about this stuff, because you must have some, because this is something quite different from what we've covered before. And as you found from the last exam, there's a lot of details here, and these details are important. So I'll be spending time expecting you to be able to tell me about those details, right? And figure out what things are important and how they're used and how things go together. Right, so exam 4 will be in some ways a recap of not a recap, a repeat of exam 3, just with different material. And I know exam 3 was tough for a lot of you. So, Rogelio, that's a really good question. That's right. So transcription is low, so it is still being transcribed, and you are still getting these proteins being produced. So as long as lactose is present, and that's the key, right? As long as lactose is present, you will still get glucose being produced, but the levels at which that happens are much lower. Now, as soon as lactose disappears, right, let's say, a bacterium makes its way onto a patch of spilled milk, right, kind of a simple way of thinking about it, right? As soon as that bacterium and all of its, clonal buddies have used up all the lactose in that milk, this whole thing gets shut down. Doesn't matter how much glucose or not is present. Once lactose is no longer present, it gets shut down completely. No lac operon transcription at all. Because there's no point. If there's no lactose as a substrate, then why bother going through all this? But as long as lactose is present and lac-I is not bound to the operator, the level of transcription will vary based on how much glucose is present. Glucose is low, transcription is high. Glucose is high, transcription is low, but still happens. Yeah, no lactose, no transcription at all. That's the big on-off switch, right? Lactose present, let's go to the races, right? Glucose is present, okay, let's just dial things back a bit, right? Because we don't need as much of this as we thought. And so you can actually think, as long as lactose is present, the levels of glucose are going to fluctuate, right? The levels of glucose might go up a bit because it's efficiently being produced from lactose. You dial it down. Glucose levels get dropped because they're being used for something, right, to make ATP. Lac operon gets dialed up a little bit. Then we get more glucose and we dial it back a little bit. And it's just this constant kind of tweaking, essentially, to get the right amount of glucose out that the bacterium needs. No, so this is a inducible system. And so in an inducible system, expression is off unless an inducer is present. So once that inducer, and the inducer in this case is lactose, once that's present, that inducer leaves, right? That's this part up here. And transcription can occur. So when lactose is not present, lac-I is bound, no transcription. Well, I mean, there's a very, 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 very little bit of it because this isn't 100% perfect. It's what they call leaky, right? So occasionally RNA polymerase kind of like looks both ways and sneaks through when lac-I happens to be looking the other way, right? But by and large, there is no transcription of this operon when lactose is not there. When lactose is there, it's full steam ahead, right? You want to produce as much of, oops, let me get that back down. No. There we go. You want to produce as much of this as you can until glucose levels start going up. So that's actually in a way, this gets back to a really kind of a fundamental issue, Rogelio. So in prokaryotes, the default state of a gene is on. But that doesn't mean that a gene or an operon is always on. What it means is if nothing is present, right, there's nothing bound to that operator, that gene will be transcribed. Right? However, that's the caveat, right? If something is bound to that operator, you're not going to get any transcription. So transcription is off. And so to get transcription, you have to get that thing off of the operator. And in the case of the lac operon, the thing that does that is lactose. And it makes sense. You don't want to go to all the effort of producing these things if lactose is not present. It would be a complete and total waste. It'd be like if you were studying for an exam in psychology when you're not taking a psychology course. Like, why would you want to do that? Right? Does that make sense? So this is, well, I mean, you could study for exam 1 of genetics if you want. It's not going to do you any good. I mean, not very much anyway. Right? So that's really, yeah, you could, but I wouldn't recommend it. Study for this one. This is the important one. And so that's really the game that's being played here by the bacterium, right? If something that isn't present, it's not going to spend the energy to produce the things to use a thing that isn't present. Because if it does, the energy that is used to do that hasn't been used to do something else. Divide, let's say, or, spinach flagellum so it can get to a new food source or something. And so all the other bacteria around are going to go, ha, sucker, and are going to busy outcompete it. And that poor little bacterium is going to die sad and alone. That's a little bit of exaggeration, but it's not going to succeed relative to its competitors. So you want this lac operon shut down completely if lactose is not present, because there's no point producing it. But when lactose is present, it's like, now we got something we can work with. Let's turn this sucker on. And it does so by removing the thing that has turned it off, which in this case is the lac-I protein. And lactose does that. But also, once you've kind of got up ahead of steam, I'm using a lot of analogies here, right? Once you get up ahead of steam, you don't really need to kind of keep piling on the coal. You can dial things back a bit, right? And essentially, once the bacterium has reached a steady state of taking lactose and generating glucose, it doesn't need as much of beta-galactosidase and lac permease, right? The things that are produced from the lac operon. So now we start kind of feathering the throttle, kind of like dialing things back a little bit so that we more closely match how much we're making of these proteins versus how much glucose we have present. And that's all done through this process by the sensing of cyclic AMP levels, which are basically a metabolic sensor. They're not a sensor, they're a metabolic readout. And so the levels of cyclic AMP are basically interpreted by the catabolite activated protein, CAP protein. So that when there's lots of cyclic AMP around, that means there isn't much glucose. And so we do need lots of expression from the lac operon. So the CAP-CAMP complex promotes transcription. Once this glucose starts going up, well, we don't need as much lactose being turned into glucose. We don't need as much of these enzymes and proteins being present. We don't need as much transcription. And so as glucose levels go up, cyclic AMP levels go down, and transcription tapers off. Doesn't disappear, it just tapers off, as long as lactose is present. Okay, so that's actually got us pretty nicely. Thank you for the questions, they're very helpful. Got us quite neatly to the end of the lac operon. Now we're going to, on Friday, and this one's a real doozy, if that makes any sense. It's a really difficult one. We're going to look at the trip operon. And the trip operon is kind of crazy, because we have the same principle, switch on, switch off, but we also have a process of something called attenuation, which is similar to the CAP-CAMP sort of deal. It's fine tuning, but the mechanism is very different, and it's actually quite hard to get your head around. So have a read through this so that it isn't completely fresh to you on Friday, and that will help a lot. Okay. All right, everybody. Thanks for being there. I shall either see you in a little bit for lab, or I'll see you on Wednesday. So have a lovely day, and I'll see you soon.